Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Here is the essay, the 1958 publication by Matthew Meselson and Franklin Stahl, entitled The Replication of DNA in Escherichia coli, stands as a cornerstone of molecular biology. This essay undertakes a detailed examination of the work, scrutinizing its claims, methodology, and conclusions, thereby offering a critical evaluation of its enduring impact and contribution to our understanding of DNA replication. The analysis will proceed in a section-by-section manner, directly addressing the arguments as presented within the original text. Section 1: Introduction and Prior Models of DNA Replication. The initial portion of Meselson and Stahl's paper sets the stage by acknowledging currently existing models for DNA replication. These models, namely conservative and semi-conservative replication, provide the conceptual landscape against which their experiment is designed. The conservative model proposes that an entirely new DNA molecule is synthesized alongside the original template, which remains intact. In contrast, a semi-conservative model posits that each strand of the original DNA molecule acts as a template for a new strand, resulting in two DNA molecules, each containing one original and one newly synthesized strand. Meselson and Stahl correctly identify the importance of distinguishing between these models through experimental means. Their presentation emphasizes the theoretical elegance of each model, avoiding any preemptive dismissal, which strengthens the subsequent objectivity of their experimental data. The strength of this section lies in its concise and impartial depiction of the prevailing scientific context. They avoid presenting any model as inherently preferable, allowing their experimental work to guide the conclusions. Section 2: Experimental Design and Isotope Labeling. The most critical element of Meselson and Stahl's experiment involves the use of isotopes to differentiate between newly synthesized and pre-existing DNA. They grew Escherichia coli in a medium containing a heavy isotope of nitrogen, nitrogen-15, rather than the usual nitrogen-14. This allowed them to incorporate nitrogen-15 into the DNA of the bacteria over multiple generations, creating a population of cells with distinctly heavy DNA. The decision to employ nitrogen-15 stemmed from its minimal perturbation of DNA structure and function, an important consideration frequently overlooked in earlier isotope labeling experiments. The subsequent shift to a nitrogen-14 medium allowed them to track the distribution of the original heavy DNA as the bacteria replicated. The experimental design demonstrates a high level of foresight, accounting for potential artifacts and ambiguities from the onset. The process of extracting DNA and analyzing its density using cesium chloride, equilibrium, density gradient centrifugation emerges as the cornerstone of their approach. The choice of cesium chloride is justified by its ability to create a stable density gradient when subjected to centrifugal force, permitting precise separation of DNA molecules based on their density. Section 3: Cesium chloride gradient centrifugation and data acquisition. The implementation of cesium chloride density gradient centrifugation constitutes a methodological marvel. Meselson and Stahl carefully documented the parameters of centrifugation, including rotor speed, time, and temperature, ensuring reproducibility by other researchers. The technique itself involved establishing a density gradient within a centrifuge tube containing a solution of cesium chloride. DNA, extracted from samples taken at different time points during bacterial growth in the nitrogen-14 medium, was then added to the gradient. Upon centrifugation, the DNA molecules migrated to positions within the gradient corresponding to their buoyant density. This allowed for separation of DNA molecules differing in isotopic composition. The critical component was the utilization of ultraviolet absorbance measurements to visualize the position of DNA bands within the gradient. By scanning the centrifuge tube with ultraviolet light, They generated a profile of DNA concentration versus density. This quantitative approach minimizes subjective interpretation of the results. The care in documenting all aspects of the procedure contributes significantly to the credibility and reproducibility of their work. They addressed the potential for artifacts arising from non-equilibrium conditions within the gradient, thereby bolstering the robustness of their conclusions. Section 4 results after one generation, and the elimination of the conservative model. The results obtained after one generation of growth in the nitrogen-14 medium proved pivotal. Meselson and Stahl observed a single DNA band located at a density intermediate between that of pure nitrogen-15 DNA and pure nitrogen-14 DNA. This single band immediately ruled out the conservative model of DNA replication. The conservative model predicted the existence of two distinct bands after one generation, one corresponding to the original heavy nitrogen-15 DNA, and another corresponding to newly synthesized light nitrogen-14 DNA. The presence of solely an intermediate band constitutes irrefutable evidence against this prediction. The clarity with which they present this finding showcases the power of their experimental design. The intermediate band strongly suggests that each DNA molecule after one generation consists of some combination of nitrogen-15 and nitrogen-14 DNA, consistent with either the semi-conservative or dispersive models. Section 5 results after two generations and the confirmation of the semi-conservative model. The subsequent generation provided definitive support for the semi-conservative model. After two generations of growth in the nitrogen-14 medium, Meselson and Stahl observed two distinct DNA bands. One band corresponded to the intermediate density observed after one generation, while the other band corresponded to the density of pure nitrogen-14 DNA. This result definitively excludes the dispersive model of DNA replication. The dispersive model predicted that after two generations, all DNA molecules would possess an equal mixture of nitrogen-15 and nitrogen-14, resulting in a single band located at a density intermediate between the density of the one generation, intermediate and pure nitrogen-14. The presence of two discrete bands, one at the intermediate density and the other at the pure nitrogen-14 density, aligned perfectly with the predictions of the semi-conservative model. Their meticulous approach in obtaining and interpreting the data permitted a conclusive determination of the mechanism of DNA replication. Section 6 Control experiments and addressing potential artifacts. Meselson and Stahl wisely included several control experiments to account for potential experimental artifacts. They tested the stability of DNA during centrifugation and addressed the possibility of DNA degradation leading to altered density profiles. They also investigated whether the presence of nitrogen-15 might somehow affect the rate of DNA replication, potentially skewing their results. Furthermore, they examined the effect of shear force on the DNA molecules, ensuring that the centrifugation process did not inadvertently break or alter the DNA, which could have influenced the density measurements. These control experiments demonstrate a clear understanding of potential confounding factors and a commitment to rigorous scientific practices. The inclusion of such controls enhances the reliability of their findings and strengthens the impact of their work, showcasing scientific responsibility. Section 7 Discussion of error and limitations. A distinguishing characteristic of Meselson and Stahl's work is their transparent acknowledgement of potential sources of error. They discussed limitations of the density gradient centrifugation technique in terms of its precision and resolution. They acknowledged the possibility of slight density variations within the DNA bands, representing small deviations from the ideal semi-conservative model. They also considered the potential for incomplete separation of DNA bands, particularly if the difference in density among the bands was very small. Recognizing these limitations allows for the identification of potential avenues for refinement and improvement in future experiments. This candid approach strengthens the scientific integrity of their work. Rather than claiming absolute certainty, they provide a balanced assessment of the strengths and weaknesses of their methodology. Section 8 Comparison to other potential replication mechanisms. Meselson and Stahl indirectly address other conceivable mechanisms of DNA replication beyond the three primary models they test, while they focus on conservative, semi-conservative, and dispersive replication. Their data also argues against a more granular form of dispersive replication, where short segments of newly synthesized DNA are interspersed with short segments of old DNA. Such a model would predict a blurring of the bands, especially at later generations, which was not observed. They do this without explicitly mentioning this, allowing the reader to deduce the broader implications. Their findings, therefore, extend beyond simply validating the semi-conservative model, providing evidence against a range of other potential replication mechanisms. This unspoken strength contributes to the comprehensive nature of their study. Section 9 Implications for Genetic Information Transfer and Mutation. The confirmation of semi-conservative replication has profound implications for understanding how genetic information is accurately transmitted from one generation to the next. This mechanism ensures that each daughter cell receives a complete and accurate copy of the parental DNA. Furthermore, the process of semi-conservative replication provides a framework for understanding how mutations might arise. If a mistake is made during the synthesis of a new DNA strand, this error will be permanently incorporated into one of the daughter DNA molecules. The semi-conservative nature of replication underlies how these errors become permanently fixed in the genome. The power of their confirmation of semi-conservative replication provided a deeper understanding of both how genetic information is preserved and how it can change. Section 10. The role of DNA polymerase and enzymatic mechanisms. While Meselson and Stahl's experiment elegantly defined the mechanism of DNA replication, they make no direct claims regarding the enzymatic machinery involved. The discovery and characterization of DNA polymerase, the enzyme responsible for synthesizing new DNA strands, came later. Their work provided the critical framework for understanding the function of DNA polymerase. It established that DNA polymerase must be capable of accurately copying a DNA template strand, ensuring high-fidelity replication. It is a subtle testament to their brilliance that a methodologically focused paper would lay the groundwork for enzymatic analysis. The study of DNA polymerase and other enzymes involved in DNA replication built directly upon the foundation that they establish. Section 11 Subsequent refinements in understanding DNA replication. Following Meselson and Stahl's groundbreaking work, subsequent research has elucidated the complexities of DNA replication in far greater detail. The discovery of Okazaki fragments, short stretches of DNA synthesized discontinuously on the lagging strand, revealed the intricate mechanisms required to copy both strands of the DNA molecule. The identification of various proteins involved in DNA replication, such as helicases, primases, and ligases, has further expanded our understanding of this process. Meselson and Stahl's initial discovery laid the groundwork for all future discovery, yet their primary claims remain central to understanding. They never overstate their findings, allowing future work to extend their results. Section 12 The impact on understanding genome stability. The semi-conservative replication mechanism has direct relevance to the concept of genome stability. The accurate replication is essential for maintaining the integrity of the genetic code. Errors in replication can lead to mutations, which can have detrimental consequences for the cell or organism. Understanding DNA replication is essential for understanding the mechanisms that cells employ to maintain genome stability, including DNA repair mechanisms and error correction systems. The foundational insight that DNA strand templates are maintained at the end of cell division provided a clear avenue to investigate the error correction mechanisms. Section 13 Techniques inspired by Meselson and Stahl's experiment. The density gradient centrifugation technique employed by Meselson and Stahl had applications far beyond the study of DNA replication. This technique has been adapted and refined for the separation and analysis of various biological molecules, including RNA, proteins, and viruses. It has also found use in the study of cellular organelles and other subcellular structures. The ingenuity of their methodological approach has inspired countless adaptations and innovations in related fields. In this way, the contribution of their research can be seen far outside the immediate world of genetics. Section 14 challenges to the model and their resolution. While the semi-conservative model of DNA replication has stood the test of time, early challenges did arise. Some researchers questioned the universality of the mechanism, suggesting that alternative modes of replication might exist in certain organisms or under specific conditions. However, subsequent research has overwhelmingly confirmed the semi-conservative model as the predominant mechanism of DNA replication in all known life forms. Occasional claims to the contrary typically prove to be experimental artifacts or misinterpretations of the data. It is a testament to the rigor of Meselson and Stahl's work that it remains the standard model despite continued investigation. Section 15 The significance of radioactive isotopes as a counterpoint. The choice to use a stable isotope like nitrogen-15 rather than a radioactive isotope represents a subtle stroke of brilliance. Radioactive isotopes, while offering higher sensitivity, introduce the potential for radiation damage to the DNA, potentially confounding the results. By opting for a stable isotope, Meselson and Stahl eliminate this concern, further strengthening the validity of their conclusions. Their experiment stands in contrast to many contemporary studies that relied heavily on radioactive isotopes, often without fully appreciating the potential for artifacts. Their careful methodology highlights a level of scientific maturity and foresight that distinguishes their work. Section 16 The role of mathematical modeling in confirmation. While Meselson and Stahl's experiment is primarily experimental, the interpretation of their results implicitly relies on mathematical modeling. The predicted distribution of DNA bands under each replication model, conservative, semi-conservative, and dispersive, can be mathematically derived. The observed results are then compared to these theoretical predictions. This interplay between experiment and theory solidifies the validity of their conclusions. The mathematical underpinnings, though not explicitly stated, add another layer of rigor to their. Section 17 The societal context of scientific discovery. Meselson and Stahl's work occurred during a period of intense scientific discovery in the field of molecular biology. The structure of DNA had only recently been elucidated, and numerous researchers were vying to understand the mechanisms of DNA replication, gene expression, and protein synthesis. Their experiment stands out not only for its scientific rigor but also for its clarity of presentation and accessibility. This allowed their findings to be rapidly disseminated and widely adopted by the scientific community. Section 18 Ethical considerations and data transparency. Meselson and Stahl's research adheres to the highest ethical standards. They meticulously documented their experimental procedures, provided access to their data, and accurately represented their findings. They did not engage in any form of data manipulation or selective reporting. Their commitment to scientific integrity serves as a model for future generations of researchers. The openness in their methodology allowed their work to be verified and extended upon by Section 19 The enduring legacy of Meselson and Stahl's experiment. Meselson and Stahl's experiment has had a profound and lasting impact on the field of molecular biology. It stands as a paradigm for elegant experimental design, rigorous data analysis, and clear scientific communication. Their work not only revealed the mechanism of DNA replication but also established a foundation for future research in genetics, genomics, and biotechnology. Their discovery continues to inform our understanding of fundamental biological processes and provides insights into the causes and treatment of human diseases. Section 20 Implications for the development of molecular biology. The Meselson-Stahl experiment is significant for showcasing the power of molecular biology by combining techniques from biochemistry, genetics, and physics. They were able to address a fundamental question about the nature of life. Their success further galvanized the field of molecular biology, leading to an explosion of research in other areas, such as gene regulation, protein structure, and signal transduction. Their emphasis on quantitative data and rigorous experimental design became a hallmark of molecular biology research. Section 21. The interdisciplinary approach to DNA replication. The Meselson-Stahl experiment underscores the importance of interdisciplinary collaboration. Both Meselson and Stahl brought different expertise to the project, with Meselson's background in physical chemistry and Stahl's background in genetics. Their combined expertise proved to be essential for the success of the experiment. They demonstrated how diverse perspectives can be integrated to tackle complex biological problems. Section 22. The human element in scientific discovery. While Meselson and Stahl's experiment is often presented as a purely objective and rational process, it is important to acknowledge the human element involved. The experiment required creativity, persistence, and a willingness to challenge existing paradigms. They faced numerous technical challenges and setbacks, but they persevered in their pursuit of the truth. Their personal qualities contributed significantly to their success. Section 23. The broader impact beyond direct genetics. The influence of Meselson and Stahl's work extends beyond the confines of genetics and molecular biology. The principles of semi-conservative replication have implications for understanding population dynamics, evolutionary processes, and even the spread of information. The understanding that information is inherited and replicated with high fidelity has parallels in other fields, such as computer science and sociology. The wide applicability of their findings demonstrates the fundamental nature of their discovery. Section 24. The question of alternative replication origins. While Meselson and Stahl demonstrated semi-conservative replication, their work did not address the question of where replication begins on the DNA molecule. The discovery of origins of replication and the mechanisms that control their activation represents another important chapter in the history of DNA replication research. Their experiment focused on the global mode of replication, leaving open the question of where replication initiates. Section 25. An ongoing process of scientific refinement. The story of DNA replication continues to unfold even today. Researchers are still actively investigating the intricate details of this process, including the mechanisms that ensure accurate replication, the role of DNA repair pathways, and the coordination of DNA replication with other cellular processes. Meselson and Stahl's work serves as a point of departure for all subsequent investigations. In conclusion, Meselson and Stahl's The Replication of DNA in Escherichia coli stands as a paragon of scientific inquiry. Through meticulous experimental design, transparent data presentation, and judicious interpretation, they provided compelling evidence for the semi-conservative model of DNA replication. Their work has not only withstood the test of time, but has also served as a cornerstone for countless subsequent discoveries in molecular biology. Its enduring significance stems from its elegance, its rigor, and its profound implications for our understanding of the fundamental processes of life. The quiet revolution initiated by this single paper continues to inform and inspire scientific exploration into the very essence of heredity.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.